Okay, great. Thanks very much to Sophion for organizing a wonderful conference in a beautiful place, and thanks so much for inviting me to talk. So I'd like to talk today about, one of the main foci of my lab right now is looking at plants to discover potassium channel targeted small molecules, and usually openers, and that's what I'm going to talk about today. So I don't have to introduce KV channels because Martin and Ian did a great job of introducing KV channels and KV7 channels in particular for Ian. And also I'm going to use mostly 2-electrode voltage clamp today, and Iris did a great job of introducing that as well. So thank you, everyone. So here we're going to talk today about two projects. One where we're looking at finding openers for KV1 channels, and another one where we're looking at finding openers for KV7 channels. And these play roles in different parts of the body. KV7s, as Ian pointed out, in neurons is very often KV7.2, 7.3, and that's what we'll be focusing on today. And for KV1s, we're going to look at KV1.1 and 1.2. They form heteromers in the CNS and peripheral nervous system as well, and usually always heteromers actually. KV1.1, probably the nervous system is not normally homomeric except in certain disease states. So the first project I'm going to talk about, guided by Native American traditional medicine practices, we've discovered a universal, as in it fixes every single ataxia-linked KV1.1 mutation we tried it on, opener from a plant that corrects EA1-linked KV1.1 mutant channels. And secondly, we're going to talk about a KV7.3 opener that's actually in a Polynesian traditional botanical medicine. So, and nothing today here has been published, and it's the first time I'm presenting any of this data. So, apart from this one slide, where last year we found the first ever small molecule that corrects any of the episodic ataxia 1 mutants in KV1.1 that I'm aware of, and it was actually gallic acid from 3 different plants. It shows up in 3 different plants: nettles, bladderwrack kelp, and Pacific 9-bark. And these were all used. The reason we discovered this was these were all used by the Kwakwaka'wakwa First Nations in the Pacific Northwest to treat ataxia. So they'd figured out that they could treat ataxia in people by using these plants, and we found out the molecular basis for that. So that was in 2023. Then another group, Servitini and colleagues in 2023, a couple of months later, found that niflumic acid can also rescue the V408A KV1.1 mutation that's linked to episodic ataxia type 1. And they found that it could also fix a mouse model of V408A as well. Niflumic acid is sort of a general potassium channel, another ion channel modulator. It's not very potent, hundreds of micromolar, but it was good at fixing episodic ataxia in vivo. And so we've kind of been following on trying to find better openers because our gallic acid only rescued 2 out of the 5 of the mutants that we tried. So ataxia, okay, what is it? It's a movement and gait disorder, causes lack of coordination, is relatively rare in its genetic forms, maybe 1 in every 20,000 people or even less, has some form of episodic ataxia. And episodic ataxia 1, which is one version of episodic ataxia, is caused by KV1.1 or KCNA1 variants, most commonly loss of function, but not always. These can sometimes be also associated with epilepsy as well. So what we were trying to do is, can we find better KV1.1 openers that not only work on the wild type, because plenty of people have found those, they have to fix the mutants. And so we look back at Native American medicine, and they very often use pine needle concoctions and pine twig concoctions to treat what we would call diseases of hyperexcitability or excitability in general. So we thought, okay, given that Native American documented treatments of ataxia are understandably rare, it's a very rare disease, and how would they identify what they were, we thought we'll go more general and look at excitability disorders. So we took a number of different samples from conifers that we've collected in the field, places like Yosemite, and also one that this kind of is a garden type conifer. This is Sawara cypress. This is lodgepole pine here we got from Yosemite. This is mountain hemlock, this is whitebark pine, and down here is ponderosa pine. And if you look, we're using 2-electrode voltage clamp over KV1.1 channels, and this is wild type, and we found 1 to 50 extracts of these. We're actually pretty good at negative shifting the voltage dependence of activation, so we get more current at the more negative voltages here, and you can see the shifts up here. So, okay, that's fairly promising. Pinus albicollis didn't really do anything, so we thought this is a good start. So what do we actually want from a Kv1.1 opener to fix ataxia mutants? Well, we noticed that quite a lot of these actually slow down the activation. That's not necessarily what you want. Even though they're increasing the tail currents and negative shifting the voltage dependence of activation, we didn't really like this property. But if you look at them, of C. piscifera at the top here, you see it doesn't slow down activation, so we were particularly interested in that one. If you look at the data summarized here, you can see that you get nice shifts in the voltage dependence of activation, hyperpolarizations, especially with these three samples, these three trees. And then if you look at C. piscifera, you can see how it doesn't slow activation, whereas all the other ones do. So we thought we focus in on a Solara cypress or false cypress, which is also called C. piscifera. So we then tried the V408A. This is all in 2-electric voltage clamp in vitro again. We've tried the V408A mutation that's linked to episodic ataxia 1 in humans, and the C. piscifera extract could actually improve the function of that as well. And there is a slight shift in the voltage dependence. It actually makes the voltage dependence align with the wild type channel as well, rather than the mutant channel in black. And if you have unclamped oocytes and look at the resting membrane potential, you can see that the C. piscifera extract causes a really nice shift to hyperpolarization, so in the resting membrane potential. So I'm not going to show the actual chemicals today because none of this has been published, but I'm just going to call it metabolite Z. So we screened a bunch of chemicals from C. piscifera, from the metabolites from this conifer, and we found that actually metabolite Z at 10 micromolar induces a really nice increase in current, especially at negative voltages in wild-type Kv1.1 currents. If you get up all the way to 100 micromolar, you get about 40% of the channels are open even at minus 120 millivolts. You get a nice hyperpolarization of the resting EM, and the EC50 for shifting the voltage dependence of activation is about 12 micromolar. If you look at for shifting the EM is 9 micromolar. Doesn't slow down activation, as we saw for the whole extract, and also it really slows down deactivation, right? So you can see how different the tail currents are, so it probably stabilizes the open state of the channel. So that's all great. So this time, Kv1.1 almost always probably exists in healthy myelated axons and in healthy brain as heteromers with Kv1.2. So we actually tried our metabolite from C. piscifera. We tried it on homomeric mutant episodic ataxia 1 channels. We tried it on heterozygous mutant homomeric Kv1.1 channels. It fixes some of the homozygous mutants that never occur in vivo. And if you make a mouse from a homozygous mutant, the mice are dead, basically. So it's only the heterozygous mice survive. It fixes most of the heterozygous homomeric Kv1.1 channels. So one mutant, one wild type, all Kv1.1. I'm only going to show you the heterozygous heteromeric Kv1.1, 1.2 channels because these are ones that exist in the patients. And we took 12 different mutants that cause episodic ataxia all the way through from this one, the N-terminus, right the way through to V408A. You can see it varying. Here's the effects of the mutants. So many of them get rid of a lot of the current. When we add back out our metabolite Z at 12 micromolar, you can see effects ranging from virtually nothing here for this one, but there's a slight increase in current all the way through to 15-fold increases in current from our metabolite Z. So it fixes pretty much, all of these actually, because the L155P barely, in this type of channel, barely changes the current amplitude. If you look at the V1 half shifts, again, this is with our metabolite, you can see the mutants. This is what they do to the actual voltage dependence of Kv1.1, the mutants in this type of channel. And you can see our metabolite left shifts every single one of those mutants, so it makes them function more at negative voltages. So this is kind of a universal rescuer of episodic ataxia 1 mutant channels. That was all in vitro. So we thought, okay, we're going to take this metabolite, we're going to make a mouse model. We made a new mouse model of episodic ataxia 1. It's E283K, which is in the S34 linker. This was discovered in 2017, reported in 2017, in an Italian family that had episodic ataxia 1, myokymia, painful contractions, and also some metabolic disorders by Ambrici et al. In 2017. So we made the mouse. The homozygous mutants are, this is by CRISPR, homozygous mutants are dead, as in the V408A mouse. We had heterozygous mice. They survive. They look fine. If you mimic the effects that were seen in that Italian family in terms of what triggers episodic ataxia, there's two things that triggered it in the family, actually three things. Diet was one, we didn't try that yet. Stress was the other one, and the other one was exercise. So first we tried exercise. So we put them on accelerating rotor rod, and you can see that the heterozygous mice fall off about 30 seconds earlier on average than the wild-type mice. If you give them an IP injection half an hour before of 1 mg per kg metabolite Z, the mutants are actually back to normal. So it fixes the exercise-induced ataxia. If you go to where they have to go on a balance beam, so ataxic mice are not very good on the balance beam, they slip a lot. You can see that here's the missteps at baseline, so without any stress, they're all pretty normal, right? Then when you give them an isoproterenol challenge to kind of mimic, it's a beta-adrenergic agonist to mimic stress, you can see that the wild types go up to about, on average, 5 hindfoot missteps crossing the balance beam, whereas the mutants have approximately double on average. You can see there's quite a big spread. We notice that some heterozygotes kind of are fine, other ones have really a lot of missteps. And then when we give them the metabolite Z from C. piscifera, it normalizes the hets. So it works in vivo at 1 mg per kg. Now, if you extrapolate that up to a human being my size and take into account the corrections for the different metabolic rate for the drug being processed, et cetera, it comes out about 8 milligram tablet for someone my size to fix what would be happening in the mice with 1 mg per kg. So I'm going to show you videos of, okay, this is a vehicle plus isoproterenol treated mutant mouse on the balance beam. So it's got the E283K mutation. You can see how it's kind of slipping, and now you can see its hind feet slip a lot on the balance beam. So it's not very good at getting across, it slips a lot. And I'm going to show you the other one, which is the wild type, just to show you what happens to the wild type. So, so it's isoproterenol, but just vehicle-treated wild type with no metabolite Z, same as this one. You can see the wild type's pretty good at getting across. They sometimes pause, they sometimes have to be kind of repositioned because they forget what they're doing or get bored and look around. So you'll see he's like, okay, I'm going to switch direction now, but that's not counted as a slip. It's just like, okay, and it keeps on going. So you can see how the mutant slipped a lot more. So now if we now treat them with our metabolite Z from C. piscifera, I don't know why the wild type doesn't play. Let me show, yeah, play it both at once. Okay. So this is now treated with metabolite Z. So the wild type is not really affected by metabolite Z, as you might expect, as I showed you from the mean data. That's on the left here. He's going along just fine, and then so is the heterozygous. So basically, the metabolite Z at 1 mg per kg, given half an hour before isoproterenol, prevents the ataxia in the mouse. So we're really excited about this because it doesn't just work in vitro, it works in vivo as well. Now, how does it work? Because it fixes so many different mutations. So we have a great assistant professor in our department at UCI, Alvin Yu, who does molecular dynamics simulation. So he built an all-atom model from the KV1.1 alpha-fold structure. And he built the whole thing, but here's just showing you the voltage sensor. There's our metabolite. And actually, you don't even have to put it in a kind of a putative binding site. You can have it in the solution. It will find its way into the binding site in the voltage sensor. So there's a glutamic acid, an arginine, and a lysine here. And the metabolite finds its way in there and is instably bound. So it's kind of co-opting the voltage sensor to act more like a ligand binding domain. And if you look at the whole, there's the whole channel, you can see that the metabolite buzzing around here, then it finds its binding site and gets kind of, gets in there stably and sticks there in a minute, or a few nanoseconds. There it goes. So it stays in there. So it's a little bit difficult to see from the nice videos, but if we just look at what happens in the still, basically, when the metabolite binds into the voltage sensor, it actually pulls on the S45 linker, kind of similar to membrane depolarization. So it's pulling on the pore domain, and we think that's probably opening it. And also, it makes the voltage sensor more compact, so we think it's more able to move through the membrane. One or both of those contributes to that amazing effect of the metabolite being able to fix all the different EA1 mutants that we tested. So just to summarize this part of the talk, there's Rian Manville in Yosemite, where we collected some pine trees, and he's the first author on that paper, which is now in review. He's done a fantastic amount of work on this project. So learning from the Native American approaches, we basically screened some different conifers. We found Saguaro cypress can fix all the mutants that we tested in vitro for episodic ataxia 1. It also fixes at pretty low concentration, 1 mg per kg, our E283K mouse model. And it works by kind of using the voltage sensor as a ligand binding domain, and that is able to basically mimic the effects of membrane depolarization and open the channel. So now we're going to, this is a metabolite which actually, to be honest, I think is not going to be toxic, because it's very similar to metabolites in extracts that a lot of people take every day. So we're going to try chronic toxicity studies, and I want to start doing clinical trials in the near future. Okay, so I want to talk about this other project as well, because I'm kind of excited about this one too. So we've got about 2,000 extracts in the freezer that we collected from, I get permits to collect plants from places like Yosemite. This is Muir Woods up in Northern California. This is me in the Kelso Dunes in Mojave Desert in California. That's Isaacs in Mojave Desert. This is Santa Cruz Island in the Channel Islands off the coast of California. And that's St. John in the Virgin Islands. So we go to these amazing places, we collect plants, we mash them up, we do methanolic extracts, and then we screen them, we'll test them on different channels. So here's one we did where during the pandemic we had to get rid of some money because we couldn't hire anyone. So we sent all our extracts to Charles River and said, can you test them on KV 1.3 and KV 7.2, 7.3? We want to find plants that are both analgesic because they open KV 7.2, 7.3, and are anti-inflammatory because they block KV 1.3, which is in T cells. And those are those plants up the top. And if you look, it's kind of interesting, right? So witch hazel comes up the top there, and fireweed, which is also known as rosebay willow herb, comes up twice in that group. So these are things that are topically applied in the States and the United Kingdom, and we always had witch hazel in the medicine cabinet while I was growing up. You put it on skin, broken skin for stings, insect bites, sores, whatever, and it makes it feel better. So we think that they work. One of the reasons that witch hazel bark works is by activating nociceptive neuron KV 7.2, 7.3 channels, and also inhibiting KV 1.3 to stop T cell activation. So there's just some of the plants I showed you in that little square up there. The mechanism that we found for this is that tannic acid is in many of these plants, and it's able to inhibit KV 1.3 and open KV 7.2, 7.3. There's Rian again when we took a hike in Santa Rosa Island for a 10-mile hike to collect plants, and we hiked and we camped on the beach. It was great, but our feet got destroyed because we're just like not built for hiking long distances with huge packs. And then there's the two first authors on this paper that's in revision on tannic acid. There's Rian Manville and Ryan Yoshimura. We're collecting in Boyd Deep Canyon, the Palm Desert in California. So what tannic acid is not going to be a good drug, right? It's about 1,700 Daltons. It kind of glues up everything in your GI tract. It's okay topically, but it's not really that good internally. You see a classic effect of tannic acid. It inhibits at depolarized voltages, KV 7.2.3. Here it is in fireweed extract, but we know it's tannic acid doing it, and it opens at very negative voltages. So that's great, and it's used in some, it's like used for post-hemorrhoid surgery, analgesia. For instance, topically applied tannic acid is used for that. And there's the KV 7.2, KV 7.3 effect. It also inhibits KV 1.3, and here's tannic acid inhibiting KV 1.3 quite nicely. We found in collaboration with Micah Hale and Yeshiva Othi next door to us at UCI that KV 1.3 inhibits human T cell proliferation, and we know it's by inhibiting KV 1.3. There's fireweed extract stopping mouse pain in the Paulick assay. So it's great. We think we found the mechanism for why witch hazel and fireweed work, or at least one of the mechanisms. But we're not looking for another tannic acid project. We're not going to inject tannic acid into mice to try and cure things like seizures, right? So, but we do want a KV 7.2, 7.3 opener. So we looked back at our screen and said, forget about all these up here. How about moving down to things that open KV 7.23 but don't inhibit KV 1.3? And we got a list in this box here. So things like Porsche tree, which I'm going to focus on, sugar bush hybrid, which I'm going to show you, and four-wing saltbush. So Porsche tree, I collected on a beach in St. John in the Virgin Islands, but it's actually very interesting because it's originally from India. It got spread through Polynesia and then onto the Caribbean. And there's a group of people called the Rotuma in this tiny island of Fiji, the Rotuma dependency, and they used Porsche tree as a folk anticonvulsant. They're kind of a little bit separated from the rest of Polynesia, and they have their own things going on. But they use Porsche tree in this way, so we got very interested in that because we found in the screen, or Charles River found in the screen, it opens KV 7.2.3, and we know that can be good as an anticonvulsant. So let me go through. Here's Porsche tree, and you see what it does to KV 7.2.3, increases the current, it left shifts the voltage dependent of activation. These are things that we want to see in a KV 7.23 opener, hyperpolarizes the membrane potential. There's our Rus ovata hybrid. This is probably a hybrid between sugarbush and a garden escape from, I collected this in Santa Monica Mountains where all the fancy Malibu gardens are alongside the actual Santa Monica Mountains. And you can see this characteristic effect of tannic acid. And that one was high up in that graph, so it's probably tannic acid doing it. Ultraplex canescens, which is I collected from Mojave Desert, is saltbush, and it does this weird thing where it reverses through zero. Saltbush has saponins in it, and I think what's happening is they're probably punching holes in the membrane, and you're getting kind of non-selective ion conducting pathways forming in the membrane. So we forgot about those two. We focused on Porsche tree. I screened 12 different compounds from Porsche tree, and you can see here I found one I call metabolite X that opens Kv 7.2.3, and metabolite Y is a little bit not quite as good. When you look at Kv 7.2.3, with 10 nanomolar metabolite X from Porsche tree, we see actually nice big effects. It shifts the voltage dependence by about minus 11 millivolts, and it's really potent. So the EC50 is 2.5 nanomolar. This compound at 100 micromolar does nothing to Kv 7.2. So it's really selective. It does nothing to Kv 7.2, and it opens Kv 7.3. Again, this is kind of an expanded axis. It opens, it's not hugely efficacious, but the potency is high. Its EC50 is 790 picomolar for opening Kv 7.3. So we think that we know it's very selective. We know it's one of the most potent Kv 7.3 openers known. We tested all these other different channels at 100 micromolar, so orders of magnitude higher concentration. It does nothing apart from opening a little bit some of the other heteromers that contain Kv 7.3. So we think maybe this is not a compound that's going to be used in the clinic necessarily, but it could be a good lead compound. So it works in mice at very lowish concentrations in a PTZ model of chemoconvulsant induced seizures. So you can see latencies of first seizure is doubled on average by 2 mg per kg of a metabolite X from Porsche tree. Funnily enough, it's not so good at high concentrations, so there might be another effect going on there. We've got to look at that in more detail. How does it open Kv 7.3? Well, metabolite X is predicted in SwissDoc to bind really tightly to this tryptophan that retigabine also uses. So I think that's how it's so potent. It aligns super well with that tryptophan there. We tested the validity of this by mutating first the tryptophan in Kv 7.3 and also in Kv 7.2 in Kv 7.23 complexes. And what we found was that you can mutate the tryptophan in Kv 7.2, you still get your effect of metabolite X. However, if you mutate the tryptophan in Kv 7.3, the effect goes away. So validating this, it's a Kv 7.3 opener that does nothing to Kv 7.2, but it's good at opening Kv 7.2 channels, the wild type versions. So we call this one beach to bedside to bench. It grows in beaches around the world. Other people before us in Fiji, the Rotuma dependency in Polynesia, discovered that this plant, Porsche tree, is good as an anticonvulsant. Then we found possibly one of the molecular mechanisms, if not the molecular mechanism, is that they have this metabolite that basically binds to Kv 7.3 channels and open it. It's one of the most potent openers known for these channels. It's one of the most selective openers known for these channels as well. So I would like to thank the people that did a lot of the hard work. There's Rian again. He gets around. He's in Yosemite this time with Katie Redford, who was a grad student in my lab a couple of years ago, who also did a lot of this work. And there's another picture of, that's Mojave Desert. So I'd like to thank Rian and Ryan, Dirk, who's a medicinal chemist, and Amy and Sonia, who are undergrads in the lab, who've done some, Amy did the ataxia. She was the blinded scorer for the ataxia work, and then Sonia's done some work on plant extracts as well. Our friends in the Department of Physiology and Biophysics at UCI, including Alvin, who does this amazing molecular dynamics with his student Lorenzo, and Shiva, Angel, and Michael Cahalan, who did the T-cell work. Tom Jeps and Jennifer, who worked with us on the Kv 7.45 openers that Ian talked about earlier. Pediatric neurologists that we work with, and then the Susan Samueli Integrated Health Institute, NINDS and NIGMS, who fund this work. And thank you to the UC Reserve and US National Park Service for letting us go to these beautiful places to pick plants. Thanks for your attention.
Thank you for your fascinating talk. I'm just wondering, when you did the KV1 and KV7 activators, were you able to validate that they're not activating other isoforms or families, especially since they might be binding to the voltage sensing domain, which I'm wondering whether it's preserved or not between the different...
Yeah, it's a really good question, so specificity. So the KV 7.3 opener is super specific. Of course, it could do lots of things that other proteins would even test, but I don't think it's doing it at sub-nanomolar concentrations like we showed with KV 7.3. So KV 7.3 is the first opener we found that opens 7.3. Sorry, that metabolite from Porsche tree is the first opener we found that opens 7.3 but does not open 7.5. So I didn't show that data because we previously published it in another plant extract paper. So it's the first one that's so selective it doesn't open any of the other KV7s that we've found anyway. I just wish it was a bit more efficacious, but the potency is really, really promising, I think. Yeah.
Thank you so much.
Great talk as ever, Jeff. It's a very quick question then. So why doesn't metabolite from Porsche tree work on 7.2?
Right, yeah.
And then the second question is, does it sensitize the effects of retigabine as it interacts with the same, or does it inhibit the effects of retigabine? Does it, interact with? So there's two
Great questions, Ian. So the first one is, why is it selective? Why is that compound selective for KV 7.3 when we know that KV 7.2 has the same tryptophan and it's S5? And we found in other studies that these compounds that are selective will still bind to the other tryptophans, but there's something else. And so we found that carnosic acid from rosemary is a super efficacious opener of KV 7.3. It shifts the voltage dependence by minus 62 millivolts. We published that last year, I think. So, and it actually stops cocaine addiction in mice. We have a paper under review. That's a long story. But so we've now done a follow-up that we're almost getting to the end of, where basically the trick's in that the voltage sensor is different in its responsiveness to binding of the tryptophan. So these compounds bind. So we found that GABA opens KV 7.3 and 7.5 channels. It binds to 7.2, 7.3, 7.4, and 7.5, and not 7.1 because it lacks the tryptophan, but it only opens 3 and 5 because the responsiveness is different. So it's a functional selectivity, not a physical selectivity. What was the second question?
Does it interact with block or enhance?
Yeah, we haven't tried that. We haven't tried that, but we have another example where we looked at openers that prefer 7.2 or 7.3. That was isovaleric acid and malatoxin, right? That were part of a folk anticonvulsant in Africa. And we found that, yes, they synergize with each other and with retigabine. So if you add retigabine, isovaleric acid, and malatoxin, the channel looks like a square wave. It's locked open even at minus 120 millivolts. So that, yes, you can exploit the heteromerization of these channels. And full disclosure, I have a patent application on that. Yeah.
The last question from Damian.
Great work, Jeff. Congratulations to Ian and Ryan and the whole lab. It's beautiful stuff. I really like these stories that you've been coming out with some years now. 3 questions. 1, that's a beautiful shirt. Where did you get it from? 2, can I be your US Virgin Islands collecting assistant? And 3, going back to KV 1.1 story, the metabolite Z, even at minus 120, I believe, was still keeping 40% at the high concentrations open. You've obviously found this binding site on the voltage sensing domain. So do you think it's literally binding in there and locking it open?
Yeah.
Yeah. Okay.
Yeah. It just mimics. Imagine what the voltage sensor would look like after depolarization. That's what it does.
And it's just locking it in position.
Yes. Then answer to your second question. What's your rum tolerance? And then the first question, I got this.
Not your rum tolerance.
I got this many years ago in England, and it's the old version, but it's the other red shirt.
Okay, thank you again.
All right. Thanks.